I'm Shamrin from Biotechnology in ESEC. So my case study is based on the introduction to cell division. So I've decided to take a real clinical case to actually understand this basic introduction. So this is not going to be very basic, and so let's begin. So this clinical case I took from my paper is a 53-year-old man. He was previously healthy, and he was diagnosed with CML, chronic myeloid leukemia. So first of all, what is CML? CML is blood cancer associated with the bone marrow, and he was admitted in the chronic phase, which means in the starting phase, and he came with the symptoms such as fatty, and he lost 16 kgs of weight, and he has came in with splenomegaly, and in his blood analysis uh, they have seen the leukocytosis marks, and he had a lot of white blood cell counts, and he had characteristic myeloid lineage cells on his smear, and the cytogenetic and molecular testing confirmed the presence of Philadelphia chromosomes, which means the Philadelphia chromosomes are nothing but the translocated chromosomes in the positions of 9 and 22, and this is what causes the BC or ABL fusion oncogene, and this gene is what gives or this gene is responsible for the CML, and this patient was initiated with imatinib as this is the first line tyrosine-kinase inhibitor or TKIs. Over time, this doesn't give proper responses, and he had more of the disease burden. So they have to give him sequential use of additional TKIs such as dasatinib and ponatinib, uh, and then he was cured, and he was able to live back to his normal life. And in this context, let's look into the introduction of the cell division. And for the cell division, the process it is the process by which a parent cell divides into daughter cells. We all know that, and it enables the growth, tissue repair, and even mostly replacement of the old cells or damaged cells. The cell division happens more often in our skin, and very less often and very slowly in our heart cells and so on. And the cycle usually is two phases: interphase and M phase. Interphase consists of G1S and G2, and this interphase is basically preparing the cell to the dividing event, and the M phase is where the dividing actually happens. The M phase consists of prophase, metaphase, anaphase, telophase, followed by cytokinesis, where the actual division happens. And now, so this is the M phase. So first, in interphase, the cells disintegrate, and in metaphase, the chromosomes align, and in the anaphase, the centrioles start to pull away the sister chromatids, and in telophase, it start to form a kind of a septum, and then in cytokinesis, two cells, it just divides, and the cellular organs starts to form again, and then it happens all over again. It goes, the one cell goes into interphase, and then prophase, and then metaphase, anaphase, telophase, and then again cytokinesis. This happens again and again and again. And for the cell cycle checkpoints, these checkpoints are literally proofreaders. This actually fascinates me very much because it is a very meticulous process. The cell division itself is a very meticulous process. If you look deep into it, maybe some mistakes could happen. It might be tangled, or something might be not happening, and it might be having some errors, and it might be having some not non-linear attachments to the spindle fibers. Anything could have happened, and if anything bad happens and it is not stopped, or if the cell doesn't go into apoptosis, a huge problem can happen to the organism. So these checkpoints check if everything is okay. If something is wrong, it triggers apoptosis and the cell dies. And if everything is okay, it allows the cell to go to the next process. This really fascinates me, by the way. And then these are the checkpoints. The checkpoints are cyclins, cyclin-independent kinases without CDKs, and these are associated with some regulatory proteins. So, in the case of CML, we have seen that BCR-ABL1 fusion protein acts as the constitutively active tyrosine kinase that drives the continuous proliferative signaling. So, proliferation is just the replication of the cell. So, the proliferative signaling must happen with this cyclins and cyclin-independent kinases. But this is so since these checkpoints, if in case these checkpoints are weak, this BCR-ABL fusion protein takes takes control, and it signals the cell to proliferate continuously without a stopping point, which results, as in the earlier case we saw, which results in the constant proliferation of the white blood cells and high white blood cells count, which leads to literally blood cancer. And this is the BCR-ABL1 protein. As we can see, the 9th and 22th pair is literally translocated, and we are getting the BCR-ABL1 protein. So, this BCR-ABL1 protein enhances the expression of the G1 phase cyclins, which means the cyclin D, and suppresses the CDK inhibitors effectively overriding the G1 or S checkpoint and promoting repeated entry into the S phase and mitosis. This results in excessive mitotic activity of myeloid progenitors, leading to clonal expansion, massive leukocytosis and splenomegaly, and systemic symptoms such as fatigue and weight loss, as we have seen in the earlier case. And from this perspective, let's see some diseases associated with disrupted cell division, and these include, as we have seen, CML and other leukemias or lymphomas, which means blood cancers, and sometimes solid tumors, and bone marrow failures, and developmental disorders. So these are the diseases. You can check the footnotes down there. And then comparative tables. Let's see the comparative table. In a normal cell division, a trigger for division must be the growth factors and the tissues need. And in dysregulated cell division in CML like cells, these are the signaling is wrong. Literally, some imposter is giving signals instead of the one who must give. If we look into like a metaphor like context. So yeah, the checkpoint enforcement in a normal cell division is like a strong the checkpoint signals, and in this kind of dysregulated cell division, it is weakened or bypassed. In the cyclin-CDK regulation is tight and very controlled in normal cell division, and it is sustained and compromised in dysregulated cell division. And in mitosis, the outcome in a normal cell division will be two genetically matched daughter cells, and in a dysregulated division, it is like clonal population. It is not just two, it is like a little population, and which gives such as in ALL case we saw like CML, and the genomic instability or aneuploidy is low and controlled in normal cell division, and in a dysregulated cell division it is increased due to checkpoint failures. And the clinical consequences simply tissue maintenance and tissue repair and replacement in a normal cell division, but in a dysregulated cell division it leads to blood cancer, splenomegaly, and even organ infiltration, which is dangerous. So that's it for today. Let's see, these are the first references where I take the um, clinical case, and then the second revision is from where I've learned about the cell division, and the third is where I've learned about the CDKs and the BCR-ABL leukemias and so on. These are the links for it, and these are the footnotes. You can check them out. Thank you.